going to start. So welcome everyone to today's Tech Talk, brought to you by the MPCCC. To begin with, I'd like to pay our respects to the traditional custodians of the lands from which we join together today, and acknowledge their elders, past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us. So these Tech Talks aim to provide a kind of informal and interactive forum for our research community and clinician scientists to discover and discuss the latest cancer research technologies and how they can be applied in a practical way. As I said, they are informal, and we should have plenty of times for Q&A at the end, where people can, there's no such thing as a daft question, and it'd be great if people can maybe just provide information about perhaps how they've been using particular technologies and novel ways in which they can be applied. So today we're very pleased to welcome Marshall Feterl from Nanostring as our guest expert. We've come to know Marshall very well over the last couple of years, really reflecting, the importance of digital spatial profiling, and this was kind of recognized by the BDI. And so I kind of took a bit of a lead role in drumming up support to establish this technology at the BDI, and we're glad to we're really pleased that in the end we chose to work with Nanostring in this space, and we've already established the GeoMx platform, and the Cosmix platform is also about to be launched as well. So we're going to be one of very few places in Australia that have both of these, and we're really very excited about the application of these approaches to both discovery cancer research as well as translational and clinical cancer research. So Marshall is a technical cell specialist for the GeoMx Digital Spatial Profiler at Nanostring for this kind of great Asia Pacific region. He did his PhD studies at James Cook University, where he looked at Toll-like Receptor activation in host macrophage. And then for his post-doctoral research, he looked at molecular characterization of drug resistance mechanisms in Mycobacterium tuberculosis. He transitioned to the commercial side of the life sciences 5 years ago, with an emphasis on technologies relating to immunology and genomics. And he's a real expert on these new digital spatial profiling platforms, and he's going to be discussing the new spatial biology solutions and the kind of expansion, if you like, of the Cosmix platform. So everyone, please kind of mute during the presentation, please, and then we can bring you all back on again at the end for some discussion. But thanks, Marshall, and over to you.
Thank you for the kind introduction, Roger, and hello to everyone. Pleasure to be here, albeit remotely. I'm currently in India today, so I'm sorry if I'm a little bit fuzzy. The jet lag's hitting me a little bit, but typically my home's in Brisbane, so it's always a pleasure to talk with everyone in Australia. So just to kind of give you a brief outline of where spatial biology sits, I think most people in Australia, kind of understand, the importance of spatial and how it plays into, discovery and really considering the evolution of genomics and where it sits. But, if you think about bulk NGS sequencing, when that, kind of launched in 2006, I remember during my PhD, that was a novel technique that was coming out. So I'm showing my age a little bit here. But with NGS sequencing, we know, with tissue, we're actually homogenizing the tissue. So you're getting sort of a broad overview of the actual transcriptomic profile that's within that tissue. We can't discern what specific cells are producing that particular signal. Fast forward to 2012 and the advent of single-cell RNA-seq technologies, which, Fluidigm pioneered, and of course, 10x Genomics took to another level. Very useful application, ones that's used quite often. But of course, you can dissociate the tissue into single cells, and then you can phenotypically profile them, according to their transcriptomic and proteomic profiles as well. Of course, when you're working with single cell RNA-seq, you lose spatial context. So you don't actually know where those cells are sitting within the tissue, right? So it's essentially a parts list of what's occurring and not really a true representation of the biology that's actually occurring. And we know that cells, they don't like to be disassociated, right? So moving forward to 2019 and really the launch and advent of the revolution in science for spatial transcriptomics, we launched our GeoMx platform in 2019. And then just at the end of last year, we started shipping the Cosmix SMI. And as Roger mentioned, you have both of those platforms, which is really exciting. Actually, one of the very few institutes in the world that have both. And that's very, very exciting. Let me just go forward. So I like to put this slide up just to give you an overview of, who Nanostring are. So, I mean, we're a research-driven company. We're scientists and engineers. And in this, on the right-hand side, this is Dr. Joe Beecham. So he's our chief scientific officer and really the developer of our technology. And I like to bring this up because he was at a conference at MD Anderson with his colleague, Gordon Mills, who's now the director of pathology at Oregon Health Services. And they were having a chat at the conference, and Joe was asking about, how can I design this, a way to look at unlimited plex of protein or RNAs in a tissue. And during that conversation with Gordon, he actually penned the original concept design for the chemistry that we used for the GeoMx. This was in January of 2015. And 4 years later, we launched the technology. But during that conversation, Gordon mentioned to Joe, he said, look, this has to work at scale. So these instruments have to work at scale, and they have to work at FFPE, right, in biobank tissue samples, because that's really the treasure trove of, patient samples and what we work with out of the clinic. And I think we've been very successful in accomplishing that. So, at Nanostring, we've been driving innovation, since the beginning. We have, in 2008, we launched our Encounter analysis system. So this is a bulk gene expression profiling system that works in FFPE tissue. That's the way it was designed, and that's the way that we designed our chemistry. And the chemistry itself is very simple. So 0 amplification, 0 enzymology. We do direct molecule counting in the tissue. Very unique. And we use an imaging-based approach to do that with the Encounter system. And that's really the foundational chemistry that we use to design our spatial platforms. So the GeoMx system, as I mentioned, launched in 2019, and we just launched the Cosmix, and also our atomic spatial informatics platform. So this is the environment that's based in the cloud, and where you're going to be able to interact with both your Cosmix and GeoMx data moving forward. All right, so last year was, pretty exciting. Cosmix was receiving a lot of attention and accolades. We were one of the top 10 innovations according to the Scientist magazine, and in Nature Biotechnology in October of last year reached the cover of our foundational chemistry paper and applications paper looking at spatial molecular imaging. So, very, very exciting to have our technology launch and to have Monash be a part of that journey as well. Funnily enough, like even outside of some of the scientific journals as well. We actually had an article written up in National Geographic, of all things, but this was highlighting Miranda Orr's work. So Miranda Orr is out of Wake Forest University Medical Center in the US. But what you're seeing here is a section of a human brain sample looking at 6,000 plex RNA. And what she was able to do is to find these senescent cells or zombie cells, which are accumulating tau protein. And of course, this tau protein is secreting toxins into the microenvironment around those cells and leading to Alzheimer's disease. And Miranda's using our platform, the SMI, and also the GeoMx to ID-specific pathways and drug targets for treating Alzheimer's disease. So we're already enabling discovery on the platform. Let's go back here. All right, so as I mentioned before, all of our chemistries, they share an in situ hybridization type of chemistry, regardless of the platform that we're using. And that's the way that they were designed. For the GeoMx system, on the left-hand side, we have content for protein and for RNA. And what we're doing here is a digital output or quantitation of both RNA and protein. So what we're doing here is we have the in situ hybridization probes that bind complementary to our target mRNA species. And on the protein side, we use a series of mouse or rabbit monoclonal labeled antibodies to ID that epitope or antigen on the cell surface. And the way that we quantitate is by capturing these known DNA oligo sequences. And these are covalently bound to either of these affinity probes via a UV photocleavable linker. And I'll talk about some interesting aspects of the GeoMx system here in just a bit. But we collect these barcodes, we run through a library prep, and then we do a digital output based on the collection of those barcodes. On the Cosmix side of things, of course, we use very simple chemistry. That's one of the calling cards of Nanostring Technologies. So for the RNA detection on the Cosmix, we have target binding probes, which are only 35 to 50 nucleotides, so very short probe sequences. We use 5 tiles for each mRNA species. We only need one of those to bind, though, in order to get a signal. So what that means is if you have highly degraded RNA tissues, and the tissues that you're likely to get out of the clinic, if there's RNA in that sample, we'll be able to find it. And we have a lot of data to support that as well. So to the target probe, we have a readout domain, and you can see that there's these sequential 4 landing pads. We go through 16 rounds of cyclic hybridization and imaging. So this is an imaging-based approach. And during those 16 rounds, we will have reporter probes that will bind to one of those landing pads. We will image that, we cleave that reporter off, wash off the reporter probe itself, and go through subsequent rounds. So in 16 of those rounds, we'll have 4 on events, and there'll be a 4-color combination that corresponds to that mRNA, along with a series of dark cycles as well. So we're actually capturing in the off events as well. And what that allows us to do, so 0 enzymology, 0 amplification, and that allows us to plex at a very high level as well. So we have a class-leading plex with our approach. On the protein side of things, of course, we use antibodies, and we have a site-specific linker built in for a readout domain. We go through a very similar process, but instead of 16 rounds, we use 8 rounds currently. We can get up to 64 plex with those proteins. If you're interested in doing a deep dive into either of these, you can look at our Nature Biotechnology papers that were launched in May of 2020 and October of 2022 last year. All right. So going back to that conversation that Joe had with Gordon at MD Anderson, that conference, we talked about scalability of the platform. So the way that Joe designed them was to make sure that they followed industry standard tissue processing. And that's what we've done. So we don't have any proprietary kind of slides. You just use a positively charged slide that you get out of the histo lab. You stick your tissue onto that slide and you go through the prep process. All right. And we have our probe hybridization workflow for both the GeoMx and the Cosmix platform. For GeoMx, we can accommodate up to 4 slides at a single time. We go through and we select regions of interest to collect those barcodes. And then we have downstream processing on either an nCounter system and or an Illumina NGS platform. For the Cosmix system, again, the system can accommodate up to 4 slides at once, but it's a standalone instrument. So there's on all of data analysis and processing is done on the cloud. So there's no downstream analysis that's required on an Illumina system, right? As I mentioned before, we also have our AtoMx platform. So this is the cloud-based solution, which is required for Cosmic workflows, but we'll be able to integrate GeoMx data into this environment as well. So all of your scaling, compute, storage can be done through that particular platform. So both platforms allow a lot of flexibility in terms of how you design your study. So the GeoMx system has a 35 by 14 millimeter scanning frame, and the Cosmix has a 20 by 15 millimeter scanning frame. But what that allows us to do is put different types of tissues onto the slide itself. So multiple tissues from mouse brains, core needle biopsies, etc. Whatever you have, as long as you can stick that tissue to the slide, that we can profile it. Another really exciting aspect or application is the use of TMAs. So if you're looking at patient match pairs, or you can get punch biopsies out of your samples, this increases the throughput on the system itself. But in terms of reagent economy as well, the more samples that you can get onto a slide, the farther you can stretch your dollar. And the cost of the probes, regardless if you're running a TMA and or a single tissue, is exactly the same. For GeoMx systems, the way that we scale those projects is by increasing the number of sampling events. For the Cosmix system itself, there's no change. So using just a single tissue or a TMA, cost of the probe is exactly the same, and there's no scaling in terms of the cost for the projects. It's just the scan time, so the area that you're scanning on the tissue itself. So that's our Nanostring ecosystem. So we have a lot of flexibility, and really the platform that you're going to choose is based on the actual biological question that you have in the tissue, right? And sometimes that can be a little bit confusing, but of course myself and we have our partners at BioStrategy that are around to help you through that journey as well. So just feel free to give me a call and we can sit down and have a chat about what you want to accomplish. But with the GeoMx system, so the GeoMx is capable of whole transcriptome analysis in up to 150 proteins, and it's a multicellular approach. So what that means is we will hone into a particular region of interest. So this can be the tumor microenvironment, for example. And say you're interested in, CD3 population. So what we do is we can label those CD3 cells with a fluorescently labeled antibody. We can mask over the cells in that region. So we need about 50 to 200 cells. We mask over that region and we can pull whole transcriptome information directly out of that and or 150 proteins. So a high degree of plex. And the GeoMx is a very fast system. So it's essentially the workhorse that you'll find out of the lab that Scott will be running there. So this is more of like a global multicellular kind of approach, right? You can think of bulk NGS kind of sequencing, essentially is what this application is. If you're interested in single cell, then you move over and migrate over to the Cosmix system, and that can get down to single cell or subcellular resolution. So if you're interested in the individual cells and the conversations that they're having, you move over to the Cosmix system. Now, because the Cosmix is based on the quantitation of those species is based on an imaging-based approach, plex is a limitation currently, although we are addressing that. So, with these in situ systems, regardless of platform, optical crowding can be an issue and spectral overlap, right? But based on our chemistry approach, as I mentioned before, so we don't do any sort of amplification of that signal. We have very small punctate reporter probes that we can do that allows us to scale and get to plex to a very high level. All right. So to give you an example of how you might use each of these, this is broadly and not necessarily cancer specific example, but here we have a mouse sagittal brain section, and we're highlighting the hippocampus, right? So with the GeoMx system, you can see this structure here. There's a lot of invaginations and folds of the hippocampus. So with the GeoMx system, what we can do is we can outline that specific structure, right? And there's no gaps in that area. We're not looking at like a gridded array approach, but we can look at the specific area that matters. We can pull whole transcriptome information out of that to look at, if we're looking for specific biomarkers or pathway analysis, we can do that with the GeoMx system. However, if you're interested in the individual cells that comprise that particular structure within the tissue, then you would migrate over to the SMI. Yep. So this is just high level in terms of sort of questions that you can ask. We believe that both systems are complementary. So with the Cosmix SMI, obviously for those of you that are familiar with single cell RNA-seq, essentially that's where these projects actually began. So we're able to segment each of these tissues into a UMAP, looking at different dimensional expression profiles to segment each of these individual cells. But from this UMAP, we're actually able to extrapolate where those cells are actually sitting in space. And we can do that in terms of plex, anywhere from 100 to 6,000, which is very exciting. So this is a new announcement that we have, and up to 64 plex protein. And the throughput is anywhere from 2 to 20 slides per week, depending on the assay that you select. For the GeoMx system is whole transcriptome for human and mouse. We have some custom content available as well, 150 proteins. And then depending on your study design and the types of samples that you're running, you can process up to 60 slides in a given week. So I'll focus on GeoMx first. As I mentioned, whole transcriptome for human and mouse. It's a biology-driven approach, so we can outline specific structures within the tissue that actually matter. So it's not just a fishing type of event. You can ID the areas of biology that actually matter to you. In terms of sample types, FFPE, fresh frozen, whole mount, TMAs, organoids, and spheroids, we've all run on the system. As I mentioned, if you can just stick a tissue to a slide, we'll be able to profile it. We're truly multi-omic, so we can analyze RNA and protein, and on the same section with the GeoMx system. And I've already talked about throughput. So I've already talked a little bit about the chemistry, so I'll just skip by that a little bit quickly. So again, we're collecting these barcodes off of the tissue. And just briefly how the workflow works. So we use imaging reagents to actually stain the architecture of the tissue. So that's to outline the specific cell populations of interest with fluorescently labeled antibodies. And the platform is completely open. So if you have specific clones that are working in your lab, if you can conjugate them to a fluor that excites in one of the channels that we use on the system, you can use that to visualize your cells. And then we also stain the tissue with our profiling content as well. So once you add both of those reagents to the tissue, you put that onto the system and it's ready to be analyzed. So this is the workflow itself. So again, you're going to stain your tissue with the fluorescently labeled antibodies in the content. We take a high resolution multiplex IF image, and then we outline regions of interest on the tissue, which are depicted by the white circles here. So what the system then does is it'll outline each of these regions of interest. It shines UV light over that area. The barcodes are then liberated off of the tissue. Those barcodes are then collected into a single well of an on-instrument 96 well plate. And that process is repeated for each region on the tissue that you identify. The barcodes are then collected, and then we run an NGS library prep and process those slides on an Illumina sequencer. Those file types are then converted into one that's recognized by the GeoMx instrument. And we have an on-instrument analysis suite that we can use to have a look at your data. And of course, you can take these files and run them through your own pipelines as well. All right. So what's unique about the GeoMx system, as well as we have these semiconductor chips that are on the system. They're comprised of these millions of tiny little mirrors. And what that allows us to do is to outline these non-contiguous regions of tissue. So right here, we have a colorectal cancer sample. The tumor cells are stained with pan-cytokeratin, and then we have a CD45 stain. But what the mirrors can do is with these fluorescently labeled tumor cells or any cell population of interest, we can create a specific mask over that area, which you can see on the top right-hand side. So we create the mask over that area, we shine UV light, and then we collect whole transcriptome or 150 plex protein, up to 150 plex protein. You can then flip that mask over to highlight the tumor microenvironment, and then we can collect information directly out of that area as well. All right, so GeoMx, it's extremely flexible. What those micromirrors allow us to do is to profile the tissue in a variety of different ways. So you can use geometric shapes, so any from circles, squares, freehand polygons, anywhere from 50 micron up to 660 micron in diameter for a circle, or 685 by 760 for a rectangle is the largest ROI. Within the geometric shape, you can apply the segmentation strategy that I just mentioned before. Your tissue itself as well does not have to be contiguous in touching. You can have dispersed cells that are dispersed throughout the region of interest. We can still mask over those. And you can apply a contour strategy as well. So you can see here on this particular example, we have equilateral tracks that are placed across the tissue. Each of these are whole transcriptome sampling events that are done. So you can look at different things like signaling pathways, immune cell infiltrates, whatever it may be. This can also be done in concentric circles as well. So you can look at a specific feature in the tissue and look at radiating patterns of RNA and protein expression in the tissue. If you want to take an unbiased approach, you can just simply place a grid over the tissue as well, and you can profile the entire area, should you choose to. So not only in cancer, I don't know if we have any neuroscientists or those working on glioblastoma or other types of brain cancers as well, but we have plenty of applications to be used in the brain as well. So just to kind of take things back to the early days of GeoMx, this was the first publication that we had in Nature Medicine. And we worked with Jen Wargo out of MD Anderson, and also with James Allison, who's a Nobel laureate. But the very first publication that we had on GeoMx actually is we discovered tertiary lymphoid structures and actually their role in response to IO therapy and melanoma. So this is the first publication that we had in Nature, which is really exciting. This wasn't even in a field, or it wasn't even a field of study until these folks discovered that using our platform. 18 months later, Jen ran another study, and she expanded upon the role of B cell heterogeneity and contribution to tertiary lymphoid structures and regulating the immune response to IO using the GeoMx. And funnily enough, at the same time, Goran at Lund University over in Europe ran an independent study at the same time and found the exact same results. So they both submitted to Nature, and we were able to have a chat with Nature about that. But this was pretty exciting because this is the first independent and orthogonal validation of the same scientific finding using the same platform. So that's something that we're very proud of. And those were both published back-to-back in Nature in 2020. Here's another example. I love to do this one. This is an older study as well. This is from David Rim's lab at Yale University in the US, but I like this because it's a nice example of how you can use the platform. But David is a pathologist, and he's trying to identify specific biomarkers that are predictive of IO response in patients. I don't think I need to labor on about the importance of IDing, whether or not a patient will respond to IO therapy here, both for patient management and also just from a cost perspective as well. So what David did is he created a TMA format. So looking at, I think, 59 different samples of patient match samples, and wanted to look at 44 different proteins across that particular tissue to see if he could ID a predictive biomarker for response. So this was his ROI selection strategy. What he did here is he stained macrophages with CD68. He used a pan lymphocyte marker, leukocyte marker of CD45, stained the melanocytes with S100B and PMEL, and then created specific masks over each of these separate populations. So what you can see here in the middle is that we're highlighting each of the individual tumor cells that are within this, this tumor biopsy here, and also the macrophage population. And just because of lack of space here, he also created the CD45 compartment as well. Excuse me. So what do you find here? When we went through an unsupervised analysis of this, the tumor cells were segmented on their own, and that the CD45 and CD68 populations were segmented in stroma. Which was interesting to find is that the clinical responders to therapy, which you can see here in blue on the top, had an increase in specific immune cell markers as well. The tumor itself was devoid of these, so this was a hot tumor, but non-infiltrated. So looking at the results here, what he found, which was extremely interesting, is that PD-L1 showed the strongest association with overall survival in the CD68 macrophage masked compartment. And B2M in the CD45 compartment was associated with both progression-free and overall survival in these patients as well. So what's interesting here is that the ability to segment between these different cell populations and really deconstruct that micro environment within the tissue is critically important to ID these particular biomarkers. So if you had used single cell or bulk or even other spatial technologies without the ability to segment, you wouldn't necessarily be able to find that particular result. I just want to highlight this as well. So for those of you that are looking at, building different collaborations or ones of your own, we have a track record for standardization of these particular studies and using large-scale multi-site studies. So this is one that we did or published in cancers, and we worked with a variety of groups across the world, including some here in Australia. But this provides best practices for working with FFP and fresh frozen tissues on both of our assays, and gives you guidance for experimental design and data analysis. So for those of you that are starting out, this is a really great publication to download, and it can give you a little bit of a path forward as well, what to expect. In terms of content, I've already mentioned that, so I'll move on from that. Just to let you know, if there are any, we have optimized protein panels as well. So these are all standardized and off the shelf for you, and the same thing with our RNA assays. But if you do have particular targets that are not included within our panels, you can spike in up to 10 probes into our protein assays, or to the whole transcriptome or cancer transcriptome, we can spike in up to 400 genes, right? And that's for any species as well. We also have something called the proteogenomics platform. So this is the ability to look at whole transcriptome and 150 proteins on the same tissue sample. So we're the only platform that's doing this at this high of plex. And you can download this and look at our white paper in order to do that. Okay. In terms of scalability with cohorts, so I'm not sure if Scott has a bond system, but we can do upstream automation for staining. So this takes the hands-on time down to 30 minutes. So that'll increase the turnaround time for him in his lab. And then we also work with Illumina, and we use their Dragon BioIT pipeline to convert our file types into one that's recognized by the GeoMx. So essentially what that means is that we can get through a lot of samples quite quickly in a week. And that's really one of the benefits of GeoMx is the scalability. We have more content that will be launching in the second half of this year. So being able to look at user-guided and AI-driven ROI selection to speed up that process. So that will be interesting for Scott and for those of you that are accessing the platform. We'll be showing an increase in protein plex here in the next couple of months. And we'll also be launching a TCR panel as well. So you'll be able to look at TCR diversity within human samples. So we've run a couple of these studies here in APAC, and we've got some pretty exciting results. This should be commercially available, I think, around September. That's very exciting. All right, second half of the talk, I will talk about the Cosmix SMI. So this is our single cell subcellular resolution platform. All right, so this just gives you a little bit of context of what we can do with the platform. So what you're looking at here is a 5 micron thick melanoma tissue, 1000 plex RNA. Each of these little punctate dots corresponds to a spatially resolved mRNA species in space, right, in a three-dimensional structure. What's cool about this here is this is, in a single run, we can process 4 million cells and almost 2 billion transcripts in a single run. So if we compare that to what was done in the the Human Cell Atlas project, right, which was across 406 labs, they profiled 26 million cells across 406 labs, and that took several years in order to get through that many cells, 26 million cells. We're doing 15% of that in a single run, and we're also adding spatial context to that. So you can just see how far the field is actually advancing and the type of information that you're going to get off of our platform. So as I mentioned before, what the Cosmix does is it provides a bridge between these single-cell RNA-seq studies that a lot of you have done now. But essentially what we can do is you can almost bypass, single-cell RNA-seq workflows and start to, move into spatial. So now we're adding not only an X and Y component, but a Z component to those cells are actually sitting in space. Right? So we can look at the distribution of those individual cells as they are throughout the tissue. Right? So how we do that? We take 8 to 9 Z stacks through the tissue at a 0.8 to 1.2 micron step, and we spatially resolve each of those 1,000 transcripts through each of those Z stacks. Right? So what you're seeing here is that 1,000 plex RNA panel on the melanoma tissue that I mentioned earlier. And as we get up to the dorsal view here, you can actually see that this is the nucleus here that's opaque, and you can see the distribution of some selected mRNA marker genes within the tissue itself. So you can turn these on and off, but you can actually ID specific transcripts within the nucleus, cytoplasm, and around the cell boundary as well. So these are the basic metrics of what we have on the platform right now. So we have the highest plex, so best-in-class plex at 1,000, and this is increasing. You can process any sample, so FFP, fresh frozen, TMA, organoid, spheroid, just like you can with the GeoMx. We are a truly multi-omic platform, and only one that can do at the higher plex level that we can do. Tunable throughput, so anywhere from 2 to 20 slides per week, depending on the assay that you choose and the area that you're scanning on the tissue. And we have a high degree of resolution, so we can get down to 50 nanometers in the X and Y direction. So even though the SMI just launched this year, it is a battle-tested platform. So we've been running projects through our technology access program over the past 2 years. Some people from Monash has actually gone through this program as well. But we've profiled over 70 plus tissue types. And because we have such a high plex panel, we're not beholden to tissue specific panels. So you can run our 1,000 plex panel. If you need to spike in genes of interest, you can do that into these panels as well. But we have a broad base of coverage, which is really exciting. And we've delivered over 130 projects to our customers, and a lot of people are publishing using that data already. So here's just some of the analysis here, or the metrics that you can see here, and the ability to get down into some regions. I'll show you that. All right, so essentially how it works as well. So we're looking at that transcript data. As I mentioned, we're resolving each of those transcripts through that Z stack. But we also do, we perform morphology marker protein staining in the tissue as well to basically define the cellular boundaries and the nucleus and the structure of that tissue. We take the protein information, we take that RNA information, and then we feed it into a cell segmentation algorithm to get accurate cell segmentation, which we believe is best in class. And then primarily, we compress that into a 2D image to do our secondary kind of analysis. So this is extremely important. So we use an AI machine learning driven segmentation approach that gives really robust detection of different sample types. And we're the only company that's actually doing this. So other companies, they have it in their roadmap, they might be working on it, et cetera, but we're the only company that's actually doing this. And it's extremely important to do accurate cell segmentation because remember, unlike single cell RNA-seq, you're working with a tissue, a three-dimensional structure. So it's important that you understand the anatomy. It's important that we define that cellular structure so we can accurately register where those transcripts are sitting in space. So if you're actually trying to define, what's occurring in the tissue and the biology that's happening, this is a very critically important step, and we do this in exceptionally well. How we do it, we publish all of our tools as an open source. So you can actually look at, the algorithm that we use is CellPose, so you can go have a look at that itself. And the data sets are completely open as well. So you can run our data sets through your own pipeline as well. And for protein detection as well, we use something called Celesta. So these are tools that were created by the academic community. Right. I talk about plex a lot when it comes to nanostring, and this is extremely important because in order to do, just basically the basics of accurate cell typing, or maybe even looking at cell type and state in the tissue, you need about 250 to 300 genes, right? That's the basic level that you can get to. But beyond that, you need to increase in plex, right? So for more biological insight, plex is extremely important. So that's what our 1000-plex panel allows us to do, is to look at different bins of biology within the tissue and help enable more discovery for you in your science. So what our panels provide is the ability to look at cell atlasing. So this is the distribution of individual cells within the tissue, looking at the neighborhood analysis, so how those cells are actually arranging within the tissue and space and how they're interacting with one another, looking at cell state and function, and then also looking at receptor ligand interaction. So we have over 100 specific receptor ligand pairs in the tissue, where you'll be able to look at specific communications that cells are having individually with that panel that we're using. And also we'll have the ability to do biomarker discovery and pathway analysis. So again, having more data from the higher plex panels is going to enable you to actually do these applications. Lower plex panels will not allow you to do that. Okay. As I mentioned before, just coming back to our chemistry, so those very, very short probe sequences allows us to work with real world FFPE samples. So in this example, this is in our Nature Biotechnology paper. We don't do any RIN scoring or DV200 scoring on our system. But in this particular example, we did, just to prove a point, but this is a non-small cell lung carcinoma data set. And the RIN values were 2.4 down to unmeasurable, with DV200 scores from 60% down to 20%. So in a lot of these samples, you wouldn't even be able to do bulk NGS, right? But with our probe sequences, we were able to capture anywhere from 94 to 98% of these past our QC metrics. Right, so degraded samples, as I mentioned, we're able to work with extremely well. Right. A lot of different groups have used this data set, so it's a publicly available data set that we have online, and they've developed particular tools to analyze our data, and people are really happy with what we'll be able to produce on the platform. This is a recent data set that we provided looking at healthy and HCC liver cancer tissue. We have a high degree of sensitivity here, so capturing over 11,000 mean transcripts per cell, a large diversity within the panel. So within that 1000-plex panel, 70% of the targeted genes are novel, as detected within the tissue, and a high degree of specificity. So we spike in negative probe sequences and false code sequences into each of our panels as a way to look at our limit of detection, and over 99% of those gene counts were on target in this data set. And of course, I've already talked to you about the importance of accurate cell segmentation, which we're best in class, and I think in doing that. We also have protein content. I think this is extremely important as well, and is becoming more important in spatial as well. So, looking at RNA and protein within the same tissue, I think is extremely valuable. This is our first panel there. So this is a 64-plex human IO focused panel. And you're able to process 1 slide per day at a 1 centimeter squared tissue area. And we'll be having the ability to add custom targets to this in the second half of the year. We'll also be expanding the content here. But this is an IO focused panel. We grew up in oncology, and this is really, the first protocol for us. So some important bins of biology be important for you. So this just relates to the scanning area. As I mentioned on the Cosmix, you have a 20 by 15 millimeter area in order to scan your tissue. So you place your tissue on there, and then you'll put fields of view over the area. So you basically just put tiles along the area that you're interested in. So you don't have to worry about the fluorescently labeled antibodies to guide ROI selection. You just tile over the area that's important to you. You press go and you walk away from the instrument. Okay. For all of our protein panels, these have all been validated as well. So we go through an initial screen with this, and we go under a pathologist review, looking at IHC standing pre and post conjugation with our barcodes. And then we run this through a variety of different cell lines, so 52 cell lines that we've analyzed. And we find a high concordance between the replicants and preparation methods. So from slide to slide, instrument to instrument, and automated sample prep variability, we get a lot of reproducibility in terms of the data that's coming out. And you should find that from your core as well. So protein obviously is extremely important here. So this provides, full endpoint functional validation of your RNA discoveries, and you can do that on the same platform. And we also have, post-translational events that we can look at on the system. And we can look at subcellular location and activation of those. And I'll show you an example here. So this is from a breast cancer trial called the SMART trial out of Oregon Health Services. And here we have a core needle biopsy. And we're looking at a specific inlay here, again, looking at that 66 protein. And particularly, we're focused on EpCAM CD45 and looking at actually the phosphorylation of ERK1 and ERK2 at the same time. So this patient right here, you can see, the individual translocation of phosphorylated ERK1 and 2 to the nucleus. This particular patient was given a phosphorylation kinase inhibitor. A biopsy was taken post-treatment. You can actually see a decrease in the MAPK and ERK signaling in this particular patient here. So these are some of the interesting things that you can use the platform for. In this particular example, this is from a Grant Kolar's lab. He's a diabetes researcher. So this is a preprint that's now been out on bioRxiv, so you can have a look at that. But one of the major applications that most people look at initially when they're using Cosmix is just cell atlasing. So what cells do I have in my particular tissue in healthy, in cancer? Are we looking at transitional states? Are we looking at pre and post therapy? These kinds of things, right? So in this particular example, as I mentioned, Grant works on diabetes, but he wanted to profile or create a cell atlas of the islets of Langerhans within the tissue, right? And within the islets of Langerhans, there's these endocrine cell populations of beta, alpha, and delta cells, and they comprise a very small portion of the overall tissue itself. But so what he did is he collected 443 or profiled 443 islets of Langerhans and ran our 1000-plex panel. To this point in time, no one had really looked at the spatial distribution of the subpopulations of the islets of Langerhans and specific endocrine samples. So what he looked at was healthy tissue. He looked at type 2 diabetic patients and also metabolically healthy but obese patients, and looked at the spatial distribution of these particular cells. So what he found here is over on the left-hand side, you see an individual with a healthy islet of Langerhans. So you can see a nice distribution of beta cells, which are predominant alpha cells and delta cells here. Moving into the type 2 diabetic setting, you can see that there's a significant reduction in beta cells, which they know. And then looking at the metabolically healthy but obese setting, you can see that these islets of Langerhans are significantly enlarged, but then there's also a complete almost obliteration of these beta cells. They also looked at some immune cell populations that were circulating around this area and looked at some interesting interactions here. But these are the kinds of things that you can look at, right? So what cells are in there, what's important to you, and how does that define the biology that you know about from other assays and from your expertise in the field. So I'll spend the next probably 5 to 6 minutes here just talking about the roadmap going forward. So at AGBT, this is a big tech conference in the US that happens every year. We've been attending since 2019. And at that time with the GeoMx, we showed 84 genes, and that was more than 20 times what anyone was doing at the time in space. A year later, we went up to 1,800 genes. A year after that, we hit whole transcriptome. Last year at AGBT, we were at 1,000-plex genes for the Cosmix instrument, and this year we showed 6,000 genes. And we haven't hit a mark, or we haven't missed a mark. Excuse me, we haven't missed a mark in the past 5 years. And Joe Beecham thinks that we can get up to whole transcriptome next year. So I'm not promising anything, but I know he's cooking some things up in the lab. But this is kind of what we have our sights set on for single-cell RNA-seq. So what that 6K means, it will be a product. So it will be available in the TAP program either in Q3 or Q4. It looks like the timelines for that are being expedited, but that represents a 6-time increase in the plex that we're doing right now. We'll also be increasing our protein plex up to 120, and we'll be having other assays that are going to be launching in the second half of the year. So 1,000-plex mouse universal panel. We're looking at human neuro, mouse neuroprotein, and also customizable options for protein as well. So in terms of throughput, what we can do for this 1,000-plex RNA panel, so you can process anywhere from 2 to 8 slides per week, depending on the scanning area. So 2 slides at 1 centimeter squared, 1,000-plex is what we're looking at. For the 6,000-plex panel, you can process 1 to 5 slides per week, depending on the area, but probably more likely 1, depending on that 1 centimeter squared area. But the 6,000-plex panel takes 1.7 times longer for turnaround time. So again, why plex matters. So this is a Vernoid plot looking at different bins of biology. So everything in green is the biology that's covered by a panel. This is from a competitor with a 248 plex panel. Everything in white is the area that's not covered. So that's 248. That's our 1,000 plex panel. So again, we've profiled this or used this in over 70 different tissue types. We've delivered over 130 projects now. And we have 16 preprints and publications coming out on this platform, so people have used it. So again, this is 1,000 plex, and this is 6,000 plex, right? So this essentially covers the entire human reactome within the tissue. So we've validated this against reference materials across 35 different independent cell lines, and we're getting excellent sensitivity and specificity. We've also embedded done a nested experiment looking at our 1,000 plex panel in that 6,000 plex panel in actual patient samples. So these are PDAC samples, actually from Will Wong's lab. And we don't see any dropout, or reduction of the number of genes that are detected from when we're increasing from that 1,000 to 6,000. So this is Will Wong. This is a study that we did with him. He published on the cover of Nature Genetics with the GeoMx paper on his PDAC samples, and we ran a 6,000 plex Cosmix panel as a follow-up study. So what you can see here is the raw images of some of that 6,000 plex panel. So you can see that these cells are just chock full of RNA. We're able to actually look at that with a scale to a very high level, and also run protein on the same slide itself. So we can do up to 8 plex proteins on the same slide as the 6,000 plex RNA. And here's some of those beautiful structures that have been created there. What's interesting as well in terms of looking at the diversity of transcripts and genes from the 6,000 plex panel, what we find is once we get up to about 50,000 cells, we're capturing about 4,800 to 5,300 unique genes per cell, right? And that's 3 logs of detection over our negative control probes and false codes. So that's roughly 2 to 3 times more than you would get out of a single cell experiment. And really, the question that you need to ask yourself is, in terms of investing your time and research dollars, is you don't really need to do single cell RNA-seq upstream from this, right? So you can run a single experiment, you get more genes, more transcripts detected on a single platform with information in space just by using our 6,000 plex panel. So you don't have to do single RNA-seq, other types of profiling spatial technologies. You can do everything right here, which is pretty exciting. I'm running out of time, so I'll just go quickly through some of this data. So we get a nice distribution of cells, and you can see that the UMAP clustering here, because of the plex level, we get really, really nice segmentation. It is excellent. In terms of looking at the biology itself, right? So right here, we'll just look at signal transduction. So we have over 407 different signaling ligands captured within this particular panel. So here we're using our Atomics platform to do the analysis. And we went forward and we looked at these specific correlation matrices to look at receptor ligand interactions. And as we move through the tissue, what we can see are these specific neighborhoods that are clustering, looking at specific chemokine and cytokine interactions. And we can actually get down to the individual cell level and see how these chemokines and cytokines are actually distributing out from the tissue itself. So here we have some cancer-associated fibroblasts and macrophages. We've got malignant tumor cells and macrophages as well. And you can see that there's a radius effect of those chemokines and cytokines that are coming out of this particular area, right? So 40 micron here, 70 microns here. And there's all of these different conversations that are happening, but this can kind of lead to deeper understanding of biology and whether or not your drug target is getting there, how cells are behaving and interacting, and what that distribution in space actually means. So last few minutes here, I'll just talk about our Atomics platform. So this is our cloud-based solution that will be coming with the Cosmix itself. So what Atomics allows you to do is do all of your comprehensive data analysis on the cloud itself. So it allows for scalable compute and storage. So you'll have a single subscription that will go to Micromon, but essentially the cost for this is roughly 10% of your overall reagent spend will be put towards the cloud. It enables easy sharing and collaboration. So you can actually send your colleagues an email. They can follow that link and be embedded in a massive, almost 1 to 10 terabyte data set, depending on what type of analysis that you're doing. And they can access this data through the cloud anywhere in the world, which is very exciting. There's a high level of security and compliance around that. So it follows all of AWS's security structure. And I've already talked about cost effectiveness of it. We've also built the platform so it will be compatible with open source analytics. So we'll be able to look at different groups using digital pathology analysis and different spatial AI and machine learning tools that'll be able to integrate it into the platform itself. So if we compare that with other competitors, so in terms of interactive image and data viewer, we can do that. That's what our competitors are doing as well. But Atomics enables quite a bit more. So one thing that's really exciting is it allows for customization and data analysis pipelines. So essentially, if your bioinformatics team is creating a particular script in R or Python, we can download that script or upload that script into our Atomics platform. You can use that as a module to integrate into your analysis, and then your bioinformaticians can go on and keep coding and doing their things. On the front end, the people in the lab can then build out their pipeline using that customization option. You can also download pipelines or modules from Bioconductor as well. So you have on-demand compute power and storage. You don't have to build out that infrastructure at Monash itself. It's a hub for multi-site collaboration and also provides long-term storage should you need it. So this just gives you a little bit of an overview of our pipeline orchestrator. So this is just kind of on the back end of a study. So each of these little modules here you can see. So we have data QC, normalization, UMAPs, PCA plots, cell typing, etc. These are individual modules that are on the left-hand side. You just simply drag and drop and click those. As these modules are being executed, you can see how they're performing in real time, right? And you can share all this information. You can provide secondary analysis where you look at transcript localization or cell segmentation can be run. And you can actually integrate multiple projects across within the platform itself, right? So that's also unique to our platform to look at multiple studies, multiple slides on the platform. Customization modules, as I mentioned, we can already do that. We can do that as well. So you just drag in those metrics from R or Python for the analysis. And then I also wanted to just pause on here for the last couple of minutes. So one of the major bottlenecks that you'll have with spatial data is the actual analysis. So once I get it, how do I actually make sense of it, right? So even for groups that are bioinformatically savvy, it can take anywhere from 4 to maybe even 12 months to make sense out of a whole slide, whole transcriptome type of analysis. So what we provide is something called a spatial data analysis service. So this is a fee for service. And what you'll do is you'll provide our team with your data from the GeoMx or the Cosmix instrument. We'll go through, we'll have a consultation with you and find out what specific comparisons or questions you want to ask within that particular experiment. We will QC your data, and then we build particular pipelines for you. And the turnaround time for that is 3 weeks. At the end of that time, we'll give you a standardized report and all of the data ready or publication ready figures. And then we also provide you with the source code. So then you can take that information, you can give it to your bioinformatics teams. We coach you and train you on how to analyze the data, but then you can take those pipelines, integrate it into Atomics or whatever pipelines that you're using so that you can scale those studies and move forward in the future. So this is really exciting and something I think that can help you, get to your publication results quicker. We'll also be expanding more content later on this year. We'll be developing, and that's the last slide that I'll have. So I'll pause there. So thanks for your time, and I'm happy to field any questions you might have.
Right, thank you very much, Marshall. That was amazing. The whole technology seems to be going from strength to strength. Do we have any questions at all? If you do have any questions, please put your hands up. I might just get started with a couple of quickies from myself.
Sure.
So I know that certainly from my lab's perspective, we want to be using this with mouse models of cancer where the tumor cells are going to be human, but the actual tumor microenvironment is going to be mouse. And so I know that there's kind of upgrades happening at the moment, which is going to give you kind of better coverage in the mouse. But just briefly with GeoMx and Cosmix, where are we in terms of mouse transcriptome?
Yeah, so GeoMx, so we've done this before in like PDX models with GeoMx. So not an issue. We do have to take serial sections.
Mm-hmm.
So on a serial section, you'll do one analysis of the humanized or the human cells or the tumor cells that you're putting into the animal, and then in the other section you'll be looking at you'll use mouse whole transcriptome to look at that content. There are some overlap in genes though, so you'll be able to kind of look at cross comparisons between shared genes in the platform that are detected in both species. but that's for the geomics. For Cosmix, currently we have content for human 1000 plex, but we will have a mouse 1000 plex universal panel in Q4 of this year.
Okay, great.
Mouse 6K will likely be sometime in 2024. I would think probably the second half of 2024.
Okay.
Yeah.
Sorry, they've got one. Oh, Dusty's got a question. I work on adult stem cells in the gastrointestinal tract using mouse models. What type of coverage for adult stem cell markers are provided in the Cosmix panel? That might be something you want to take offline, but can you give any insight to that, Marshall?
I don't know specific stem cell markers that we have, Dusty, but if you can just shoot me an email, I'll send you a copy of the gene list, and you can circulate that in the group. And as I mentioned, you can spike in anywhere from 7 to 50 genes into our 1000 plex panel, and or we can do custom up to 350 plex.
With
I probably didn't get to mention that, Mike. I apologize for that.
Bit of a, hopefully it's not a dumb question, but with the kind of upgrades that you're talking about, increasing the plex and so on, are they all compatible with the technology that we've installed here, or do we need to upgrade the equipment that we now have on site?
Yeah, no, that's a great question. So as I mentioned at the top of the talk, right? So we built these to scale. So any improvements that are made are going to be done via chemistry or software.
Right.
So there's not going to be a reason to buy a new system.
That's good.
Yeah. It should make you feel better. Yeah, you can spend your money on reagents.
Yeah. And then just one other question I had. I think the bioinformatics, in the cloud is a great idea in terms of data sharing and so on. Then I also saw that you were providing the analysis service, but just taking that a stage further, we have one group in particular here who is applying artificial intelligence to histopath images.
Mm-hmm.
We've already been discussing about being able to apply this to much more complex spatial data sets, because, I think a good example was that immunotherapy study that you highlighted, where they were looking at biomarkers of response to immunotherapy. I mean, sometimes that can fall out with like 2 or 3 markers, but often the actual biomarker signature is much more complex and almost impossible for the human eye to observe, and you need kind of AI approaches to actually tease out those signatures. And so I was wondering whether Nanostring were, working with people in the AI space to actually add AI to the analysis pipeline in terms of the data analysis, rather than the segmentation and so on.
Yeah, that's a great question. And to answer your question, short answer is yes, we are. So we formally started working with Visiopharm. I don't know if you're familiar with them. So they're an AI software company that works in the digital pathology space.
Right.
So we're integrating that into our geomics analysis workflows. But essentially what we can do is use their software to use H&E images and the multiplex IF images taken off geomics, marry that with the WTA data, and use that for feature identification and biomarker ID within the tissue itself.
Okay.
So I find the AI space is really exciting. There's a lot of groups that can do some really interesting things, and we're certainly keen to collaborate as well and share best practices and thoughts about how we look at the data, and maybe that can help.
Well, I might. This is
software.
Yeah, this is Jan Ying Song from my department, and so I might put him in touch with you guys, and because there may be some interesting projects that you could, we could do on a collaborative basis maybe, because as you just indicated, it's such a rapidly changing field. It would be good to kind of get in there whilst the iron's hot, as it were.
Yeah, absolutely.
Cool. Okay. So if there's not any more questions, I just want to thank Marshall one last time for a really fantastic presentation, and thank everyone else for their attendance. Our next tech talk is next week on the 25th of May with Tara Mack, who's dialing in from Tennessee. So we've got a very much very kind of international program at the moment. And that presentation is going to be about curating clonal hematopoiesis, crikey, of indeterminate potential in human genetic data sets. So very interesting talk there, and we'll send out information about that in due course. But I just want to thank both Marshall and you all for attending, and yeah, we'll look forward to seeing you next time. So thanks, Marshall.
Thanks, Roger, and thanks, Hannah, too, for setting this up. Really appreciate it.
Thanks very much.
Everyone. Yeah. Cheers, everybody. Have a good day.
Thanks, everyone. Bye.
All right. Bye-bye.